Anatomy of lens. We already have a video in our channel on the dimension of the crystalline lens. Please watch that before beginning this video. I have linked it in the description box. Let's move on to the anatomy of crystalline lens. The crystalline lens is an avascular, transparent, elliptic structure that helps in focusing light rays on the retina. It is located in the posterior chamber, anterior to the vitreous and posterior to the iris. The lens is suspended from the surrounding ciliary body by the zonular fibers. The ciliary muscle contraction can cause a change in lens shape, increasing the power of the eye, which allows the near object to be focused on the retina. This is an image showing the focus for a distant object. This is an image showing the focus for the near object. Note the shape and curvature of the lens in both the images. This mechanism that causes an increase in lens power is called accommodation. The lens is located in a cup-shaped depression called the patellar fossa. The posterior lens surface is attached to the anterior vitreous face by the hyaloid capsular ligament, which is a circular ring of adhesion. Within this ring is a potential space, which is an area of non-adhesion between the lens and the vitreous, which is called the retrolental space of Berger. Let's see some important areas of the lens. This is a cross-section of the lens showing the equator, the anterior pole and the posterior pole. The structure of the adult lens is determined during the embryologic development. The lens vesicle, which is the first lens-like structure observable in the developing embryo, is composed of a layer of epithelial cells that forms a hollow sphere. The posterior cells differentiate and elongate, forming the primary lens fibers. As these fibers grow and reach the anterior cells, the center of the sphere fills. Thus, the adult lens has no posterior epithelium because it was used to form the first lens fibers, otherwise called as the primary lens fibers. During the rest of the life, cell division occurs. The cells that forms elongate to form the secondary lens fibers that are laid down outer to the all earlier fibers. Let's move on to the histology. Lens capsule. The lens capsule is a transparent envelope that surrounds the entire lens. In this image, the capsule is shown in black, which covers the entire lens. Its thickness varies with the location, which is thickest at the pre-equator region and thinnest at the posterior pole. The capsule consists primarily of collagen. It contains no elastic fiber, but it is highly elastic because of the layer-like arrangement of the fibers. The capsule encloses all the lens components and helps to mold the shape of the lens. The zonular fibers insert into the capsule, merging with it in an area from the equator to near both the poles. The lens capsule provides some barrier function, preventing the large molecules such as albumin and hemoglobin from entering the lens. Lens epithelium. Adjacent to the anterior lens capsule is a layer of cuboidal epithelium, the anterior lens epithelium. These cells secrete the anterior capsule throughout the life and are the site of metabolic transport mechanisms. In this image, the epithelium is shown in blue, which is absent in the posterior side. As seen previously, this is because there is no posterior epithelium as it was used during the embryological development to form the primary lens fibers. The basal aspect of the epithelial cell is adjacent to the capsule and the apical portion is oriented towards the center of the lens. The lateral membranes of the epithelial cells are joined by desmosomes and gap junctions. Let's see how new lens fibers form. The band of cells in the pre-equatorial region that lies just anterior to the equator is called the germinal zone. This is where the cell division occurs. The cell division continues throughout the life. As each cell divides, a daughter cell migrates posteriorly towards the equator and it withdraws from the cell cycle and forms into a new lens fiber. Each newly formed cell elongates. The basal aspect stretches towards the posterior pole and the apical aspect towards the anterior pole. Let's see a single cell. Here, the apical and basal parts are shown. This process occurs all around the equator with fibers stretching towards the poles from all aspects of the lens periphery. As the cells in each layer elongate, the cellular nucleus move with the cytoplasm. A line drawn to connect the dots of these nucleus would have an arcuate shape towards the anterior aspect, and the configuration is called the lens bow. Eventually, as it loses all the cellular organelles, the elongated cell becomes a lens fiber. The structure of the lens is similar to an onion. Each layer of fibers approximates a layer of an onion. Lens fiber production continues throughout the life, with the new lens fibers being laid down outer to the older fibers. All fibers formed from mitosis in the germinative zone are called the secondary lens fibers. The cross-section dimension of a fiber are approximately 3 to 9 microns. Each fiber has a long crescent shape with the broad sides parallel to the lens surface and the narrow sides at an angle to the surface and located near the lens poles. Lens fiber cytoplasm contains a high concentration of proteins known as crystallines. This is the reason why the lens got its name as crystalline lens. This protein accounts for approximately 40% of the net weight of the fiber. The distribution and concentration of the crystallines contribute to the gradient index of the lens. The crystalline concentration varies from approximately 15% in the cortex and 70% in the nucleus. Divisions of the lens. The primary lens fibers from the elongating posterior epithelium form the very center of the lens, the embryonic nucleus, and all subsequent lens fibers are laid down outer to the core. In the beam of slit lamp, these are seen as zones of discontinuity. Depending upon the period of development, the different zones of the lens nucleus includes the embryonic nucleus. It is the innermost part of the nucleus which corresponds to the lens up to the first 3 months of gestation. It consists of primary lens fibers which are formed by elongation of the cells of the posterior wall of the lens vesicle. Fetal nucleus. It lies around the embryonic nucleus and corresponds to the lens from 3 months of gestation till birth. Infantile nucleus, which corresponds to the lens from birth to puberty. And adult nucleus, which corresponds to the lens fibers formed after puberty to the rest of the life. Cortex. It is the peripheral part which comprises the youngest lens fibers. As the lens fibers reach the poles, they meet with other fibers in the layer, forming a junction known as a suture. The anterior suture is formed by joining the apical aspect of the fiber, and the posterior suture is formed by joining the basal aspect. The secondary fibers formed during the embryological development meet in 3 branches, forming a Y suture. The anterior suture is an upright Y shape and posterior is an inverted Y shape. As growth continues and the lens becomes larger, the suture becomes asymmetric and dissimilar. The limbs of the anterior and posterior sutures are offset, and the complexity of the suture contributes to the lens transparency. Zonules of Zinn. The lens is attached to the ciliary body by a group of thread-like fibers called the zonules of Zinn or the suspensory ligament of the lens. The lens fibers belongs to the category termed microfibrils. The fibers arise from the basement membrane of the non-pigmented ciliary epithelium in the pars plana and from the valleys between the ciliary processes in the pars plicata. Most fibers attach to the lens capsule at the pre-equator and the post-equator regions, and few attach to the equator directly. The zonules are interwoven into the components of the capsule. Those that attach to the lens are known as the primary zonules. Secondary zonules join the primary zonules with each other or connect processes to one another or to the pars plana. And tension fibers anchor the primary zonules into the ciliary valleys to form a fulcrum. Lens transparency. The factors that play a significant role in maintaining the lens transparency and clarity are as follows. The avascularity, the tightly packed nature of the lens, the arrangement of the lens proteins, semi-permeable character of the lens capsule, the pump mechanism of the lens fiber that regulates the electrolyte and water balance inside the lens. Nourishment. The lens is metabolically active and requires nourishment in order to maintain its growth and transparency. By 9 weeks into human development, the lens is surrounded and nourished by a net of vessels, the tunica vasculosa lentis, which is derived from the hyaloid artery. Beginning in the 4th month of development, the hyaloid artery and its related vasculature begin to atrophy and completely disappear by birth. In the postnatal eye, the clockwork canal marks the former location of the hyaloid artery. After regression of the artery, the lens receives all its nourishment from the aqueous humor. Thanks for watching. Subscribe to Let's Learn Optometry for more optometry and eye care videos. And please don't forget to like and share this video. Thank you.